A very good evening, everyone. This is your session 2 with me. So, in the previous session, we have discussed about the week 1 content and we have practiced few MCQs related to that. So, similarly, in the session 2, we will be discussing about the week 2 content and we will be solving few MCQs related to the week 2 content. So, I am your TA for the session. My name is Dipanshi, and now let's start with the session. So, in this session, we will be covering few topics related to week 2, such as genes and pedigree, Mendelian pedigree patterns, which we have also discussed in the session 1 in the week 1, and we have practiced few questions related to Mendelian pedigree patterns. So in this session also we will be continuing with few problems related to Mendelian pedigree patterns, and then we will look into the complications to the pedigree patterns. We will also study DNA cloning and hybridization techniques, in which we will be dealing with the vector-based cloning, nucleic acid hybridizations, and PCR-based DNA analysis. So let's start with our practice problem 1. So in this question, the question says it is the schematic representation. In this diagram, it's shown here represents a segment of genomic DNA harboring a gene and the corresponding mRNA transcribed from this region. Various elements of the gene, such as intron, exon, transcribed region, and the start and stop codons are indicated. Three functional segments of the mRNA are marked as A, B, and C. Which one of the following options is incorrect with regard to the three functional elements identified in the mRNA? So now you have to look into this diagram in this figure. You have to tell if this is the DNA and these darker elements represent the exons and these lighter elements represents the introns, and these constitutes the mRNA, which is shown here. So you have to tell what the ABC regions depict, and out of these you have to tell that which option is the incorrect representation of these three functional elements in the RNA. So if you have any idea, then you can answer. Otherwise, I'll tell you the solution. So anyone wants to answer to this question? Okay, so if no one wants to answer, then I'll tell you it's a very straightforward. So first of all, let's see what does this region signify. So you see that from all the exons, it only represents the exonic segments in the mRNA, which means that this mRNA is spliced, because generally whenever a DNA transcribes, then it also contains the introns in it. And then this splicing happens, and which converts the pre-mRNA into the mature mRNA. So in this, they have only shown the exonic regions. So these three are coming from the exons. So this 5' region till AUG, it represents the exonic part, which will not code for the protein, as it is before the start codon, and the translation only starts from the start codon. So this will be the untranslated region, which is also known as 5' UTR. And from AUG to UAG, this will be the coding part, which will code for the protein sequence. And after this region, till the 3' end, it is the 3' UTR region, which will not code for any protein. So C will signify the 3' UTR. Now let's look into the options. So first option says that A, B, and C identify the exonic regions of the gene. So yeah, it is true. Now option 2 says A, B, and C identifies the 5' UTR coding segment and the 3' UTR of the mRNA respectively. So yeah, it is also true. Now third option says A, B, and C identify the 5' cap region, exon, and poly A tail of the mRNA respectively. So now this is not correct, because it is saying that A represents the 5' cap region. But we know that capping happens at the 5' end, but it is a post-transcriptional modification which happens after the mRNA has been synthesized from the DNA. So the DNA will not carry this cap region. So if you see these red dotted lines, so these says that this entire fragment is coming from this segment. So this A can never be the cap. Whereas if you see this C and C region, then it is also coming from the DNA, and it will not be the poly A tail, because poly A tail is not present in the DNA. It is a feature which is added to the mRNA after it has been transcribed. So it is a post-transcriptional modification. So in this way, this option is the incorrect representation for these elements. And let's look at the option 4, which says that all the three segments represent the exonic regions of the gene. So it is also true. So hence, The correct answer to this question will be the option C. That is the third option. That is ABC identifies the 5' cap region, exon, and poly A tail of the mRNA respectively. So if you don't have any further doubt and if it is clear, then now let's move to the another question. So now this is the question number 2. So what does it say? That in a field survey of sunflower species, Ram identified a new variety for the flower with the violet petals and named it X. As the genotype for this trait was unknown, he has employed the following four methods in order to understand the dominant or the recessive nature of the trait displayed by the X. Based on your understanding, identify the option that lists the correct method. So now they are saying that Ram has identified the new variety. So in which the flower, for the flower which shows the violet petals, so he don't know the genotype. So he want to identify that unknown genotype, although he has the information about the phenotype of that flower. So now what do you think which test he should perform to get the information about the genotype of the unknown phenotype flower? So sorry, unknown genotype information for the flower which has the phenotype information with him. So the first option says a backcross between the species X to a genetically similar species, a backcross between the species X to another species with an unknown genotype, a test cross between the species X with a known homozygous recessive genotype, and the fourth option says a test cross between the species X with a known homozygous dominant genotype. So now you have to reply which cross he needs to perform. So I hope you have some idea about the back cross and the test cross and when we perform this. Okay, so now let's see its answer. So its correct answer will be a test cross between the species X with a known homozygous recessive genotype. So now we generally perform this test cross when we don't have the information about the genotype and we have the information about its phenotype. So in that case, to identify the genotype of that unknown species, we generally cross it with the recessive genotype so that we can see the effect and we can trace the genotype. So I'll give you the representation for this test cross. So here you can see generally the test cross is used to study the dominant character inheritance. So we can identify a dominant character by performing the test cross. And in this unknown genotype is crossed with the known homozygous recessive genotype. So for example, here you can see. So if it would be a dominant type, then you will be seeing its, you can predict that what would be the genotype of this particular unknown species, what would be the genotype. So you can tell either it is a dominant form or it is a heterozygous form. And then you are crossing it with the recessive form. So in this way, we can perform test cross whenever we don't have information about the genotype of some species. But this can only be performed for the plants as it cannot be done for the humans as they have small size of families. And this generally for human, we perform the pedigree analysis. So with the pedigree analysis, we can study the inheritance of different traits in the human species. So we'll be studying the pedigree patterns. So as discussed in the previous lecture as well, so in this also we will be seeing few Mendelian inheritance patterns. So in the pedigree. So pedigree is nothing but it is the recorded ancestry or lineage of a person or family. So there are 5 different types of Mendelian pedigree patterns, which are autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and Y-linked. And these are the symbols which I have shown you earlier also. I think you might be aware about this, that what does this symbol signifies. And all the 5 types has characteristic features and I think you are aware about these. So we will not study them in depth. We will just perform few problems related to them. In the previous session also, we have discussed many problems related to these pedigree patterns. So now I hope you all are familiar with these. So let's proceed further. Now this is the question which you have to answer this time. So now let's see what does it say. So it says that consider yourself a genetic counselor and review the pedigree given. Nisha and Satish are expecting their first child. This disease has not affected anyone in Nisha's family. Given these, what is the probability that Satish and Nisha's child will have the disease? So now you have to focus on this line that they are saying that no one in the family of Nisha has ever affected with this disease. So now by looking at this pedigree, you have to predict that what is the probability that if Satish marries Nisha, then their child will have the disease. So now look carefully and answer me. You can answer in the chat if you don't want to speak, and I'll check. Okay, so some are saying 0%. Okay, anyone else wants to try? Okay, so now let's see. So if we look, then this Pankaj is affected. Okay, so now for Pankaj to be affected, we must know that what would be the genotype of them. So here you can see that Reshma is unaffected, whereas Pankaj is affected, and both of their parents were unaffected. So this kind of inheritance pattern, it mostly occurs when it is a autosomal recessive kind of inheritance. So both are the carriers for the disease trait, and they are giving this to the Pankaj, and Reshma is a carrier. So she is not showing any phenotype. Now, if as their offspring is also affected, so Satish should also be a carrier as he is not showing the disease. So if he has both the dominant alleles, so then this their daughter would have been the carrier. But as she is affected, so which means that Satish would also be a carrier, and he is not completely normal. So in this way, Satish is a carrier, and their daughter is affected with the recessive type. So now if Satish marries Nisha, so what do you think what would be the genotype of Nisha? So this line says that no one in her family was ever affected with the disease, which means that she would never be a carrier. So which means she would have both the dominant alleles. So if now they would have their children, then nobody would be having the disease. They could either be the carriers, but they would not show any disease, which means that correct answer will be 0%, as this trait is autosomal recessive, and hence Satish will be the carrier, but Nisha has the normal genotype. Hence, none of their offspring will ever show this disease trait due to the recessive nature of the trait. So now let's move to the another question. So this question says the below given pedigree shows an autosomal dominant inheritance pattern. Based on this information, identify the option that correctly mentions the genotypes for the individuals marked as 1, 2, and 3 respectively. So now you have to look at this pedigree and you have to tell what does 1, 2, and 3 signifies. So these are the given options. These are the given genotypes for these. Now you have to answer. And in this, they have also mentioned what kind of inheritance pattern is there, that is autosomal dominant. So it's quite clear from the question only. So anyone wants to answer? Okay, some people are saying option 1. Okay, someone else? Okay, now let's see what would be its answer. So, as it is quite clear from the question that it is a autosomal dominant inheritance, so which means that even if a single um, allele is present in the dominant, means even a single dominant allele is present, then also it will cause a disease. So, which means for an individual to be unaffected, it should carry the recessive pattern. So, with this, its genotype would be small b, and for every unaffected individual will carry this genotype small b. And if you look here, this one, it would have the means one allele would be dominant and one would be recessive. So why we are not considering both the alleles to be dominant? Because one of their offspring is unaffected. So if it would be both alleles would be dominant, then all of the offsprings would show the disease as it is a autosomal dominant. So in this way, its genotype would be capital B, small b. So now if you look carefully, then you will see that this will also be the carrier. And why we are not saying it as capital B, capital B? So in this case, you are very clear that it won't be capital B, capital B. But if you look here, then why it is not capital B? Because here also two offsprings are unaffected. As it is autosomal dominant, so it would never have the dominant, both the alleles as dominant, because if both alleles would be dominant, then all the offsprings would show the disease. So in this way, it would have the genotype capital B, small b. Similarly, the 3, which is unaffected, will have the genotype small b, small b. So the correct option will be option 1, as you have also said. Yes, so this option is correct. And I have already explained. So you can go through this when I'll share the slides with you. So now let's move to the another problem. So here is a family affected with the rare skin disorder, which does not affect the survival or fertility. Review the pedigree and identify the mode of inheritance. So you have to look into this pedigree and you have to identify the mode of inheritance. So in this, they are saying that it is a rare skin disorder and it does not affect the survival or the fertility. So you have to tell whether it is autosomal dominant or it is autosomal dominant with incomplete penetrance, it is Y-linked or it is X-linked. Okay, so do you want to answer? Okay, somebody in the chat is saying it's autosomal dominant. Okay. Okay, some people are saying it is X-linked inheritance. Very good. Anyone else? Okay, now let's see. So in this, you can get the idea from here that means if you see generation 1 and generation 2, then nothing uniqueness you find. It's not clear because the offsprings are both males and females as well, which are affected. So now if you carefully observe from generation 2 to generation 3, then the thing which you can observe is that whenever a father is affected, then all its daughters are affected, but none of its sons are affected, which means that male is transmitting its disease only to the daughters and not to the sons. So what does it mean? So it happens only in the case where it is a X-linked dominant allele, because only fathers only passes their X chromosome to the daughters and not to the son. And why it is dominant? Because even if the mother is normal, but still the daughters are showing the disease phenotype. So which means that it is a dominant in the kind of inheritance, X-linked dominant inheritance. So hence the correct answer will be option 4, which is X-linked inheritance, as affected males transmitted the disease to all his daughters, while affected females transmit to both the sons as well as daughters, because the mothers, they have the X chromosome and they pass this X chromosome both to their sons and daughters. So in this way, it is a X-linked inheritance. So I hope now it is clear that why its answer is option 4 and not option 1. Because if it would be autosomal dominant, then father would also pass this to their son as well, not only the daughters. So now let's see another concept. So as previously we were discussing about the Mendelian pedigree patterns. So in Mendelian pedigree patterns, we were focusing on the DNA that is present in the nucleus. But other than that, there is another kind of inheritance pattern which exists, in which we consider the mitochondrial genome. So we know that other than nucleus, two other organelles are also there which carries the DNA. One is mitochondria and another one is chloroplast. So mitochondria is present in the human. So we will study this. So mitochondrial genome and mitochondrial inheritance means that mitochondrial DNA will have certain disease genes, then it would be passed to their offsprings only from their mothers, so not from their fathers. That's why it is also known as matrilineal inheritance or maternal inheritance and frequent heteroplasmy. So in this way, here you can see that this is the unaffected father and affected mother. So all of their offsprings are affected, but their degree of disease can vary. Whereas here there is affected father and unaffected mother, so none of their offspring is affected. So from this it is clear that only mother passes on their mitochondrial genome to their offsprings. So, and one more thing you must know, that as we know that the DNA present in the nucleus is linear, but the mitochondrial DNA show the properties related to prokaryotic DNA, that is circular in nature. So now let's solve questions related to this mitochondrial inheritance. So the question says, given below is a pedigree affected with a late onset myopathy, caused by defects in a gene located in the mitochondrial DNA. What is the probability of individuals identified as A and B in the third generation developing the disease later in life? So in this, they have given you the pedigree and you have to tell this is a mitochondrial DNA. So the gene is present in the mitochondrial DNA. So this means that it is a maternal inheritance pattern. And you have to tell that what is the probability that A and B will have or will generate the disease in the later stage of their life. So these are the given options, and it's very clear. You can simply tell its answer, as we have just now discussed about the mitochondrial inheritance. So you can give it a try. Okay, you can type your answers. Okay, so you are saying option two. Okay. Anyone else? Okay, so now let's see. So as they are saying, it is a mitochondrial inheritance. So mitochondrial genome, any disease or any disease gene or any disease that lies on the mitochondrial DNA will only be passed from the mother to their offspring and not from the father. So in this way, the probability of A carrying the disease would be 100%, but B would be 0%. So the correct option will be option 2. And you are already familiar with this kind of inheritance as it is maternal inheritance only. So now let's see few complications to the pedigree patterns. So other than mitochondrial inheritance, there are other complications also which exists and which makes the things very complicated. So one such complication is incomplete penetrance. So it happens when you see that the phenotype skips a generation in the autosomal dominant trait. So it will carry the genotype, but it will not show any phenotype. So in that way, it becomes difficult to trace. Then next complication is the variable expression, which means that phenotype can vary from severe to moderate to asymptomatic. Then comes complementation, which means that mutation in two different genes rescue the phenotype. So if one gene has undergone some mutation, then other gene also undergoes some kind of mutation to rescue the phenotype. So it will manage the effect, and hence you will not be able to trace that there is some mutation because you will be able to see the phenotype even if there is a mutation. That is known as complementation, where both the genes are complementing each other's expression and they are means rescuing the phenotype. Then fourth one is the imprinting disorders. So we have also discussed during our session 1 that what are imprinting disorders, where one copy of gene from either parent is silenced and only the single parent is transferring their chromosome or their particular gene to the offspring. So we can say that it is the uniparental disomy. So its examples were Prader-Willi syndrome and Angelman syndrome, depending on the which of the parental gene is being silenced or which parental chromosome is getting silenced. So on basis of that, it's Prader-Willi or Angelman. Then comes anticipation. Anticipation means with every generation, the disease onset age decreases and the severity increases. So in anticipation, you can consider an example where the repeats are present. So with each generation, the number of repeats were also increasing, due to which for the next generation, the disease will start early and it will be severe as compared to its predecessors. So the next complication is the mosaics and the chimeras. So mosaicism means whenever you observe the somatic mutations which cannot be inherited, but still you observe the phenotypic patterns in the individual, such as skin color patches. Then comes the chimera. So in the chimera, what happens is an individual with two or more distinct or unrelated genetic lineages. For example, in the heterochromia, you see that a child is born with the two different eye colors. So in that way, it is a chimera in which two or more distinct and unrelated genetic lineages are present. Then comes the monogenic and polygenic effect. So with its name, it is clear monogenic means when single gene is involved. So whenever a single gene determines a single trait, it is known as monogenic inheritance. And whenever more than one gene determines a particular trait, then it is the polygenic inheritance. So now let's move to the question. So the question says, given below is a pedigree that shows incomplete penetrance. with an early onset phenotype. The pedigree shows dash mode of inheritance with dash penetrance of the disease phenotype. These are the given options, and this is the pedigree. So you have to answer this question. What do you think? What kind of inheritance pattern is this? And what is the penetrance percent? Do you have any idea what is the how to calculate the penetrance? Okay. Okay, so some are saying it is option 3. That is autosomal dominant and it is 3.3. Okay. Okay. So as it was present in your assignment 2 also, this same similar type of question was present. So let's see what would be the correct answer and why. So before moving on that, let's focus here. So here you see that in the generation 1, individual 1 was affected and the mother was unaffected. And if you look at their offsprings, then only 2 was affected. And then in generation 3, again offspring 2 is affected. But if you see here, 5 and 6, they both appears to be normal. But their offspring are showing the disease. So which means that this 5 is not showing the phenotype, but it might carry the genotype. There is some kind of possibility associated with this. And we also know that incomplete penetrance is the feature of the inheritance pattern where it is autosomal dominant. So you should be aware of this, that whenever certain trait is autosomal dominant, so even if a single dominant allele is present, so then also it is affected. So as these both are unaffected, then how is it possible that their offsprings get affected? So which means that this individual 5 is showing the incomplete penetrance for this disease phenotype. So in this way, the correct answer will be the autosomal dominant mode of inheritance. Now the question arises how to calculate the penetrance. So to calculate the penetrance, the formula is that you have to keep all the number of penetrants in the topmost side. So penetrant means which all are showing the phenotypes. And then you have to divide it with the sum of known penetrants with the penetrants. So it means that all the individuals which carries the genotype. So in this case, the individuals which there are 5 individuals which are penetrants. So 5 will be divided by 5, and this individual in the second generation, that is individual 5, he is showing the he is not showing the phenotype, but he is carrying the genotype. So hence he is showing the incomplete penetrance. So that's why we need to calculate this also in the total number, because it is also carrying the genotype. So it becomes 4 divided by 6, and its number will be 0.833 when we'll convert into percentage, and we'll get 83.3%. So in this way, it is the correct answer. That is this. Okay, now let's move to another question. So here it is given that examine the image and identify the option that correctly represents the condition of having two different eye color. So these are the options: a metabolic disorder, having more than one genetic lineage in the body, somatic mutation, and all of these. So do you want to answer? Because it was also present in your assignment too, and by the time you have also received the answers to these questions. So if you remember, then you can answer. Okay. Some are saying option B. Okay, somebody has answered option D. Okay. So somebody has written that this is the heterochromia in which there is more than one lineage present in the same body, and it is also a somatic mutation due to which this heterochromia arises. But I am quite unsure about the metabolic disorder. Okay, no problem. So as per the assignment, the correct answer was the option 2, that is having more than one genetic lineage in the same body. So it is a condition which represents this two different eye colors. But if you look into this disease, that what does it is known as? So this is means we cannot call it as a disease. So it is a condition which is known as heterochromia. That means there are two different eye colors which exist. So if you say which option of them correctly represent this, so it means by looking at this, you cannot say it is a somatic mutation. Means okay, and it is means by looking at this figure, is not a metabolic disorder. But by looking at this figure, you can say that it shows that more than one genetic lineage present in the same body. But yeah, if you talk about heterochromia, then yeah, it can arise due to the somatic mutations. And if we talk about the metabolic disorder, then there is another disease, that is glaucoma, which is also the cause of this heterochromia. Sometimes in certain people, if the glaucoma is present, then also it leads to the um, change in the eye colors, due to which heterochromia could be present. So in this way, we can say that option 2 is the correct. But yeah, we cannot ignore the somatic mutation, and that glaucoma is a metabolic syndrome. So it can also be regarded as a metabolic disorder, but not clearly it is a metabolic disorder, it is a metabolic syndrome. So in this way, its correct answer is still a confusion. You can say it is all of these. But as per the question, if you have to select any one of the option, then it would be option 2, which correctly represents this condition. So now let's move to another question. So next question says, which one of the following options is true with regard to polygenic and the monogenic disorders? So we have just now discussed about the polygenic and monogenic disorders. So option 1 says monogenic disorder more often affects the younger population, while polygenic disorder affects the older population. Option B says the mutant alleles for the monogenic disorder are inherited from parents, while the mutant alleles in the polygenic disorder represent epigenetic changes. Option 3 says while the disease causing alleles are exclusively present in the affected having a monogenic disorder, such alleles for a polygenic disorder can also be found in the normal population. And option 4 says only one of the parents contribute the mutant allele for a dominant form of the monogenic disorder, whereas both the parents contribute multiple disease-linked alleles in the polygenic disorder. So what do you think would be the correct answer to this question? It is quite clear from its name only, and we have just now discussed, so it would not be a difficult question for you. Anyone? Okay, somebody is saying option D. Very good. Others can also type in the chat. Okay, so now let's see what would be the correct answer. So the correct answer is option D. Why? Because monogenic disorders means monogenic, that is single gene is involved, whereas polygenic means more than one allele is involved in a particular disease. So hence this option is correct, that only one of the parents contributes the mutant allele for a dominant form of the monogenic disorder, whereas both the parents contribute multiple disease-linked alleles in a polygenic disorder. So this is the correct answer for this question. Now let's move to the another part, which is related to DNA cloning and hybridization techniques. So in this, we will discuss about the DNA cloning. It is also the content part in your week 2. So what does DNA cloning means? So cloning, as you are aware of the name clone. So clone means when you construct the identical copy of something. So it is written DNA, that means you are creating the identical copy of the DNA, and not just single copy, but multiple copies are made of the single DNA and identical copies of the DNA sequence. And when we perform this cloning, then we don't go for cloning the entire DNA sequence, but we only clone a particular sequence of interest, which is also known as the target DNA. And when we increase the copy number, that means increase the number of identical copies, so that process is known as amplification. And for this process, there are different things that are required. And one such thing is that is required is DNA polymerase, which is required for the replication. So replication is the process where one DNA makes its identical copy inside the cell. So for that, polymerase is required for the replication process. And this cloning can be performed either inside the cells, known as in vivo method, or in the lab or in vitro method, such as PCR. So when we perform it within the cell or by in vivo methods, so the process of generating a genetically identical copy of a cell or an organism, that is known as cloning. It produces a large number of individuals with the identical DNA. So in this, as it is within the cell, so within the individual will divide and multiple identical DNAs will be produced. It will produce DNA copies in vivo. It requires an organism and it is important for the production of proteins, antibiotics, and vaccines. So, when our purpose is to design the proteins, antibiotics, and vaccines, so then we will use this in vivo cloning. And when do we use in vitro cloning? So, as it is a cell free method, that means it is occurring in the lab. So, it is a laboratory technique for the rapidly producing or amplifying millions to billions of copies of a specific segment of DNA. As if you see in the DNA cloning in the in vivo systems, then this is a very time consuming task as it is occurring inside the organism. But if we talk about PCR, then it can generate multiple copies in a very small time fraction of time. It can produce million of copies of a specific DNA, and it produces DNA copies in vitro. And for that, it requires the tagged DNA polymerase for the amplification of DNA. So when we talk about in vivo conditions, so there are already enzymes and different reagents and different nucleotides that are required for this process already present inside the organism. But in this, as we are performing it in the lab, so we have to provide these reagents externally, such as tagged DNA polymerase for the amplification. And along with this, we have to provide with the primers and DNTPs and many other things. And we have to maintain the conditions which are there in during in vivo. So when we use the PCR, so we use it. It is important in the diagnostics, forensics, and genetic testing when we are expecting the results faster. So in this, we will only get the results. We will only get the DNA, multiple copies of DNA. So this we cannot use if we want to generate the proteins or we want to generate the vaccine. So for that, we require the cloning inside the host or in vivo cloning. So this is the difference between in vivo and in vitro cloning and why we use a particular type of cloning method. So I hope it is clear. So now let's see, with the help of this figure, we'll be understanding. So whenever we perform in vivo DNA cloning, then the specific feature of this in vivo cloning is that in this we use restriction enzymes to digest the DNA to obtain the sequence of interest or the target DNA. So restriction digestion is performed, and then restriction digestion is performed of both the vector as well as the DNA. So I hope you have the idea about what is vector. So vector is nothing, it is just a vehicle and it can attach the sequence of interest to it. And this entire segment is known as the recombinant DNA. That means the vector is nothing but it is also obtained from the organism, that it is also extra chromosomal part. For example, in bacteria, the plasmid part is considered or it is used as a vector. It is a vehicle which can take this foreign DNA and it can further transfect it into the another bacterial genome and where it could replicate and perform it multiple numbers. So in this way, this vector will also be cleaved with the similar restriction enzymes, and in that way, both will generate the sticky ends, and those sticky ends are complementary to one another. They will ligate, they will bind, they will base pair with one another, and further we will ligate it to the vector. And now it vector contains the ori, that is origin of replication, where the polymerase can bind and start its replication. So in this way, it will produce multiple copies inside the bacterial cells. So this is the clone of bacterial cells. In this way, its number has amplified, its number has increased. Whereas if we talk about the PCR, then in PCR, this is the region of interest. So we provide it with the different primers. So the primers are specific to the region that lie near to the gene of interest, so that only the region which we desire, that only gets uh, replicated or amplified, and not the other part get amplified. So for that we require primers, and in this the polymerase, DNA polymerase is provided. So most specifically in PCR we use Taq polymerase, that is also a DNA polymerase obtained from Thermus aquaticus and which is very thermostable. So that's why it is used. When we will study PCR in detail, then you will know that why do we use Taq polymerase. So here you can see it is occurring in this tube. Multiple copies have been produced of this particular segment of interest. So now let's see what are the restriction enzymes. As I've already discussed, the restriction enzymes has a particular characteristic feature, that is there are different types of restriction enzymes 1, 2, and 3. Mostly in this case, we study about the type 2 restriction enzymes, which cleaves at the recognition site. So these restriction sites have certain recognition sequences or recognition sites present in a particular protein or particular molecule or particular DNA segment which they recognize and cleaves. So these type 2 cleaves at particular places in those recognition sites. For example, it can produce the blunt ends. So when we say blunt end, so blunt end means where there is equal dissection or equal cut is produced, where both the 5' and 3' regions are equally cut to one another, they are equal to one another. And most of the cases, these two sequences are palindromic sequences. So palindromic means they are read from the similar from the forward as well as backward. So if we read these two sequences, this from forward, then it will similar to the another sequence that is complementary to this from the backward direction. So these are the palindromic sequences, and then it can produce blunt end and it can also produce the sticky ends. Sticky ends means whenever the overhangs are generated, they can either be at the 5' end or the 3' end. So in this way, they are named as 5' overhangs or the 3' overhangs, depending that at which end the uh, overhang is present. So here it is at the 3' end, hence it is the 3' overhangs. And these are the examples of the restriction enzymes. One is ALU1 that is obtained from the Arthrobacter luteus, and this name is derived from the genus as well as species name. So first letter of the genus name is taken and then two letters from the species name are taken. This one represents the serial number. So in this way, it has got its name as ALU1. Similarly, BAMH1 has got its name from Bacillus amyloliquefacans H. So B from this genus and AM from this species and H from here, and then 1 is the serial number. Then comes the PST1, which produces 3' overhangs. It is obtained from the Providentia stuarti. So in this way, it has got its name as PST1. And here I have shown you that these are the two molecules, and this is the palindromic sequence. Now both have undergone restriction digestion. So it can either produce the sticky end or it can also produce the blunt end. So the advantage of sticky end is that they can further, they are as the sticky ends are complementary, so they can further bind. So in this, you can consider that if molecule A, in the previous case, if it is your DNA of interest and molecule B is the one that is in the vector. So in this way, you can see that this particular part can bind with this particular part as both have the similar sequence. So they are complementary to one another. So in this way, recombinant DNA can be produced with the help of restriction enzymes and their generation of sticky ends. So this is the reason that how the restriction enzymes helps during the production of recombinant DNA. So why do we need to perform the DNA cloning or the hybridization techniques? Because we can generate the genomic DNA libraries as well as cDNA libraries by performing DNA cloning, and this will help us to generate the information and further we can study. So it would be helpful in the analysis, and with this we can know that what are the genes that codes for a particular protein, or what are the genes that are responsible for particular disease. So in this way, we can also means it is very helpful to understand the significance of that particular genic segment. So when we talk about genomic DNA library, so how it is constructed, we have the genomic DNA and we isolate this DNA from the cells, and then we have the plasmid vectors that has obtained from the E. coli. We perform digestion with the restriction enzymes. We ligate with the DNA ligases, both of them, and we generate a recombinant DNA. We insert into the bacteria where it could get cloned, where it could get replicated and produce multiple copies. And then we isolate this DNA and the collection of genomic DNA library is obtained. So in this way, this particular genome is further obtained in higher number. Similarly, if we perform cDNA library, then in cDNA, what happens is we do not use the genomic DNA, but we use the mRNA of an organism. We reverse transcribe it and produce the cDNA from it. So cDNA is the coding DNA that is obtained from the mRNA, and then we perform its. So mRNA is reverse transcribed to the cDNA, and in this is the cleaved cDNA with the restriction enzymes. Similarly, vectors are also cleaved with the restriction enzymes and they are ligated to produce the recombinant DNA, and further they are inserted into the E. coli and DNA is isolated. So what is the difference between the cDNA library and genomic DNA library? As it is reverse transcribed from mRNA, so mRNA don't have the intronic segments. That means it will have the collection of the cDNA, that is actively transcribed genes. So the genes that are actively transcribed, that means it will only carry the genes that have been transcribed, and it will not carry the region that is non coding or the intronic part. It will only carry the exonic region of the gene or the genome, the DNA. So this is the difference between both of them. So now let's look into the PCR. PCR stands for polymerase chain reaction. So as I have talked earlier also, that PCR is very helpful to produce the multiple million billion identical copies of the DNA of interest. And as it is quite quick, so we perform this during the forensics or disease diagnosis. So what happens in this? It involves three steps. First step is denaturation, which occurs at the 95 degrees centigrade, that is very high temperature. So in this, we denature both the strands of the DNA. So in the PCR, we need to recreate the similar conditions which are in the in vivo conditions. So that in actually when we talk about the in vivo replication, so in that there is an enzyme that is helicase which generally unbinds the double-stranded DNA. So if we have to denaturate the DNA in the vitro, so we use higher temperature because we know that at high temperature both the strands of DNA denatures, they separates, and then we have to perform the step 2, which is annealing. So for that we reduce the temperature from 95 to 55, and now in the annealing, the primers, we provide the primers because we know that DNA polymerase can't initiate its replication, and we require the primers so that they can provide a free 3' OH group, so with which DNA polymerase can allow the nucleotides to bind and complementary bind and then uh, replicate. So in this way, in step 2, primers will bind, and in the step 3, which is known as synthesizing phase, so it happens at 72 degrees centigrade. So now Taq polymerase will bind, and now it will allow the nucleotides, the complementary nucleotides, to come and bind to this primer, and then it will elongate. So in this way, the process will begin, and multiple new strands will be produced. Similarly, we will repeat these cycles for 20-40 times. So with each round of this PCR, there will be exponential increase in the number of copies of DNAs that will be produced. So this is all about the PCR reaction. Now let's solve few questions related to this.
Now let's move to this question which says, given below is the vector map of the plasmid PCZAC, the size of which is 20 kb. The relative positions of the restriction sites on the plasmid are indicated on the map. You have to set up the following two restriction enzyme digestions for the plasmid in test tube A, digestion with the restriction enzyme BAM-H1, that is single digestion, and in the second test tube, that is test tube B, digestions with the restriction enzymes with VAM-H1 and EcoR1 double digestion. You have to identify the correct option that provides the DNA fragments that you would visualize for tube A and tube B if you were to separate the digested DNA in an agarose gel. So now in this question they are saying that you are given with the two test tubes. One is test tube A and another one is test tube B. So now they are saying that in test tube A there is only a single enzyme, that is BAM-H1, so there will be single digestion, whereas in test tube B they have provided you with the two restriction enzymes. One is BAM-H1 and another one is EcoR1. So now you have to identify. So in this figure they have given you with the positions where these enzymes digests or produces the cut. So now you have to visualize this, and you have to tell that what would be the number of DNA fragments that would be produced and what would be their length. So now these are the given options, and you have to see this and you have to answer this question. So it's quite clear, you can guess. You can try to draw it with the paper or pen, and then you can see what would be the length of different fragments. As it is a circular DNA, so it's quite clear that how many number of fragments it would generate depending on the number of cleavage sites that are present. Okay, so is any guess? Okay, few people in the chat are saying option B. Okay, now let's solve this. So in this, as you can see, in the test tube A they have taken only BAM H1, and here you can see there are 4 sites where BAM H1 can cleave. So if BAM H1 cleaves, then how many fragments it will generate? One of 2 kb if you see from this to this position, then one fragment of 6 kb from this position to this position, another fragment of 4 kb from this to this, and at last it will generate the fragment from this position to this. So it will be the sum of 4 kb plus 4 kb, that would be 8 kb. So there will be 4 fragments, as there are 4 cleavage sites and each fragment will be of length. So one fragment will be of length 2 kb, another of 6 kb, third one will be of 4 kb, and fourth one will be of 8 kb. So whenever you will separate in the agarose gel, then it will be separated on the basis of its length. So how many fragments will you observe or visualize? So there will be 4 fragments or 4 bands you can see in the DNA gel, agarose gel during the electrophoresis. Now let's move to the test tube B, which says that it has used BAM H1 as well as Eco R1. So when both the enzyme digests, so there are total 5 cleavage sites. So how many fragments will it generate? 5 fragments. So now let's see the length. So one fragment will be of 2 kb, another will be of this 6 kb, the third fragment will be of 4 kb from here to here. Now Eco R1 is digesting between these 2 cleavage sites, so it will generate 2 more fragments of 4 kb each. So hence there will be 3 fragments which will share the length of 4 kb. So whenever you will visualize them during the agarose gel means electrophoresis in the agarose gel, then you will see that as their length is same, so they will produce only single band, but its intensity may vary as compared to the other bands. So you will visualize 3 bands, one of 2 kb, another one of 6 kb, and third band of 4 kb. So in this, which option is correct one, which tells the correct number of fragments and correct length of fragments? So the correct option is option B. So you were right. So now I hope it is clear that why the option B is correct for this. So now let's move to the another question, which says that which one of the following statements is not true for the restriction endonuclease. So first option is they make the double-stranded cut within their recognition sequence. Option B, they always leave 5' overhangs. Option C, they cut the strand of the DNA at the palindromic sites. And option D, any of these. So now you have to tell that which option is not true. So we have already discussed about the restriction endonucleases, that they are very specific in their cleavage, especially the type 2 restriction endonucleases. They have specific recognition sequences, and these recognition sequences have a property that in both the strands they are palindromic to one another. So in this way, this option C is true. Now let's come to the option A, which says that they make the double-stranded cut within their recognition sequence. So yeah, it is also true, because we have seen many restriction endonucleases, they were cutting the double-stranded DNA and they have a recognition sequence. So option A is also true. So now you are left with option B and option D. So option D says any of these, and option B says they always leave 5' overhangs. So from here only you can see that, as I've earlier discussed the recognition sequences as well as restriction digestion, so I have told you that they can generate the blunt ends as well as the sticky ends, and sticky ends can be categorized into 2 types. One is 3' overhangs and another one is 5' overhangs, and depending on the position at which terminal the overhang is present, so due to which we provide it name as 5' or 3'. So it is not always the case that always 5' overhangs will generate. So in this way the option 2 is not true for restriction endonucleases. And here I have attached the figure that what does a palindromic sequence means. So palindromic site means which, if read from this terminal to this, then also it is similar to the 5' to 3'. So means if you read it from here to here or from here to here, it will be exactly same. And in this way restriction enzymes 1 and 2 are cleaving it. So both have different patterns of cleavage. So one is producing the sticky end overhangs and another one is producing the blunt end with no overhangs. So in this way the option B is the correct option. So I hope it is clear that why option B is correct explanation for this question. So now let's move further and see this question number 13th, which is saying that you are setting up a reverse transcription polymerase chain reaction, that is RT-PCR experiment, to amplify a part of the coding region of a gene using the cDNA as the template. When you look at the results, you find that the PCR reaction fails to amplify any DNA. To identify the possible reasons for this result, you checked your lab notebook regarding the reagents you have used for the RT-PCR. Which one of the options given below is likely to result in the absence of any amplification? So to simplify this question, they are saying you are performing some experiment to perform the reverse transcription reaction in the PCR. Now you have given the reagents, but you see that when you obtain the results, then you do not observe any amplification. And you have noted down the important reagents for this process in your lab notebook. So now you have to check what you have used and what was mentioned in your lab notebook to check that what is that thing which is very important and which you have missed, or due to which amplification was absent. So these are the given options. So in every option, the part 2 that is written in the Roman represents what was written in your lab notebook. So don't worry, we'll read. So option A says that you used reverse primer, nucleotide bases and reverse transcriptase for the RT reaction, and what was written in your lab notebook: forward and reverse primers, nucleotide bases and tag polymerases for the PCR. Now option B says forward primer, nucleotide bases and reverse transcriptase for the RT reaction, and what was written in the lab notebook: forward and reverse primers, nucleotide bases and tag polymerase for the PCR. Now option C says random primer, nucleotide bases and reverse transcriptase for the RT reaction, and the what was written in the lab notebook is exactly the same as previous, that is forward and reverse primers, nucleotide bases and tag polymerase for the PCR. Option D says oligo-DT primer, nucleotide bases and reverse transcriptase for the RT reaction, and what was written in the lab notebook: forward and reverse primers, nucleotide bases and tag polymerase for the PCR. So now you have to tell that in which of the option there is a huge difference which has affected the amplification. You can again read these options and tell that what is the important part for this RT PCR and which they have missed. You can answer in the chat also. Okay, so now I'll tell you the answers. So the correct answer is option B. So due to option B, the amplification was not observed. So now let's see what is the possible explanation for this. So in this, as you can see in lab notebook, forward and reverse primers, nucleotide bases and tag polymerases were written, but in this what you have used, you have missed the reverse primers in this, so due to which you could not observe the amplification. So in this it is written that a gene specific reverse primer, random primer or oligo-DT is very essential to create the cDNA of the transcript of interest, and in the absence of it, the template for the gene specific sequence will not be created. So you must remember that if you want to amplify the part of the coding region of a gene using the cDNA as the template, so which means that cDNA is formed from the which you can say which strand of the DNA. So when it comes to the when we move from DNA to RNA, then one strand, that is known as the antisense strand, which converts to the mRNA, and that can ultimately undergo reverse transcription to the cDNA. So in this, if antisense strand is getting, if we want the amplification of the antisense strand, then we must require the reverse primers instead of forward primers. So reverse primer is very important part for this RT reaction, and other than reverse primers, we can also use the random primers or oligo-DT primers. So in this option 2, we have only used the forward primer, due to which we will not be able to observe the amplification of that cDNA. So hence option B will be the correct option for this particular question. I hope it is clear that why this with the forward primer is involved in the absence of any amplification and not the other two. Okay, so now let's move further. Now this is question number 14, which says both the linear and the circular form of a plasmid DNA has 3 sites for the restriction enzyme NHE1. How many fragments would you anticipate for each of the two forms, that is the linear form and the circular form respectively, following the NHE1 digestion? So now you have to answer this question. It's quite straightforward and very clear that how many number of fragments would you observe. So in this, one is linear form of plasmid DNA and another is circular form. And there are 3 sites for the restriction enzyme NHE1. So it's very clear. You can guess. You can give your answers in the chat if you don't want to speak. Okay. You can think and you can answer. Okay, so many are saying option B is the correct option. Okay, let's see which option will be the correct. So yeah, you are right, option B is correct. So why it is correct? Because if you see a linear plasmid DNA, then if there are 3 restriction sites, then it will generate 4 fragments. One from here to here, another from here to here, another from this position to this, and then this to this. So there will be 4 fragments. Whenever any DNA or plasmid is linear and there are 3 restriction sites or n number of restriction sites, then number of fragments it will generate will always be n plus 1, because it is linear and both of its ends are exposed. So one extra fragment will be generated to that of the number of cleavage sites. So its answer will be always be n plus 1. Whereas if we talk about the circular plasmid DNA, then the number of cleavage sites and the number of fragments you observe will always be equal. So in this, as there are 3 sites of restriction digestion, so number of fragments that it will generate will be 3. So correct answer for this question will be option B, that is 4 and 3. Now let's move to another question, which says which one of the following options is incorrect with regard to the cDNA library. So we have previously discussed about the genomic libraries and cDNA libraries, and we have also discussed that how the both of these libraries are different from one another. So now by keeping those points in mind, you have to see which one of these option is incorrect. So first option is helps in predicting the proteins that the gene codes for. Option B says helps in identifying alternatively spliced transcripts. Option C says helps in the identification of disease-causing mutations. And option D says cDNA of not all genes might be present in a cDNA library of a specific tissue. So now what do you think? Which option would be the incorrect with respect to the cDNA library? And I've told you a very peculiar feature of the cDNA library when we compare it with the genomic library. So with that only you can answer this. Okay, so some people are saying option C and there are some people which are saying option D. Okay, so you have confusion with this. So let me resolve your confusion and let me tell you the correct answer. So the correct answer or the correct option is option C, which means helps in the identification of disease causing mutations, because you already know that these genomic DNA libraries or cDNA libraries, so they provides you with the cDNA information, and cDNA is the DNA which is produced from the mRNA, and you know that the mature mRNA lack the introns which are not coding for the proteins. So hence it provides you information about the genes that code for the proteins. So option A is the correct explanation for the cDNA library. So this will not be the option here. Then comes helps in identifying the alternatively spliced transcripts. So I hope you are aware about alternatively spliced transcripts, and if you are not, then I'll just explain in brief that as the name suggests, alternatively and then is spliced transcripts. So it means the RNA which is spliced, and it is spliced alternatively. For example, let's assume there are total 5 exons present in a transcript or mRNA or pre-mRNA if you take. So if there are 5 exons, then its splicing can occur in different ways. There are multiple types of splicing. So in for example, in a one particular type of splicing, exon gets skipped. So if you have 5 exons, then in 4 exon gets skipped, and you get a transcript with the 1, 2, 3 and 5 exon, and you get another transcript in which exon 3 is skipped but all other exons are present. So it will have the exons 1, 2, 4, 5. So in this way, these both are the alternatively spliced transcripts. So as in this, you are looking at the mature mRNA that has been converted into the cDNA. So you will observe them differentially. So you can identify them that yes, these are the alternatively spliced transcripts. So in this way, it will also help you in identifying that. Then comes the fourth option, which says that cDNA of not all genes might be present in a cDNA library of a specific tissue. So first of all, you must know that these expression of the particular genes or transcripts, it is very specific. It is tissue specific. So not all the genes get expressed in a particular tissue. And in this they are saying, as we know the difference between the genomic library and the cDNA library. So cDNA library is based only on the genes that have been converted into the mRNA or transcribed. So in specific tissue, only specific set of genes get expressed, only specific set of genes get transcribed. So you won't see all the genes in a specific tissue. So in this way, this option is saying that cDNA of not all genes might be present in a cDNA library of a specific tissue. So it is true, you won't see all the genes, right? So in this way, this option will not be the incorrect with regard to cDNA library, it is correct. So in this way, the correct option for this question is option C, which means helps in identifying the disease causing mutations. You can see mutations, but you don't know it is a disease causing or not, because it just tell you about the sequence and you don't know either it is a disease causing or not. So it does not tell you about disease causing mutations. So in this way, this one is the incorrect with regard to the cDNA library, and hence we will select it. Now let's move to the another concept, which is DNA electrophoresis. You have heard so many times about the electrophoresis. So as its name suggests, electrophoresis, so it means in the presence of electric charge, whenever something separates, so that is known as electrophoresis. And in this, it is related to DNA electrophoresis, which means you are allowing DNA to move in the presence of the charge or electric charge, and you will see the fragments of DNA, they will be separating on the basis of their size. So you also know, let us assume that there is a tunnel and you have to pass through that tunnel. You have different things in your hand, and the tunnel size is, for example, it's 1 feet tall, 1 feet is its height. So if you are passing something from it, the smaller the size of the particular thing that you are passing through, the faster will it pass. And when you will see the larger size of a particular thing, then it will be difficult for it to pass. So similar is the concept of the DNA electrophoresis. In this also, the fragments which are smaller in size will flow faster, and the bands, means the fragments that are shorter in size, will move faster as compared to the longer bands. So in this way, you will observe different bands in the gel. So this is all about the electrophoresis. You provide the charge, and then there is a solution, a buffer and a gel in which you put the DNA samples, and then in the presence of this charge, these samples will move in this gel, and each gel has some pores, so it will not transfer to this side, it will move straight in its lane. So this is all about DNA electrophoresis. Now let's move towards another technique, that is restriction fragment length polymorphism. As the name suggests, restriction fragment length polymorphism, which means that there will be different restriction fragments that would be generated, and on the basis of their length you will observe the polymorphism, which means that there is differences that are occurring on the basis of the restriction fragment lengths. So in this, how to perform this RFLP analysis? So firstly, from all the individuals we collect the blood samples, and then we will extract the DNA from the white blood cells. So one fact I want to make here is that whenever we talk about the cells from which we have to isolate the DNA, so mostly we can use any of the cells, but the exception is that we cannot use the mature RBCs. So because mature RBCs lack the nucleus. When they are immature, then they have nucleus within them, but as soon as the RBCs mature, they lack nucleus, so we cannot use the mature RBCs to isolate the DNA. So hence we use the mature white blood cells to isolate the DNA. And as soon as the DNA is isolated, we perform restriction digestion. We add restriction enzymes in that. So somebody was asking that how to isolate this DNA from white blood cells from the blood. So this is generally done using the centrifugation methods. In that, the blood sample is centrifuged at particular RPM, and then the component of the blood which are heavier will be obtained in the form of a pellet at the bottom, and another components which are lighter, they will be floating in the topmost region, which is known as supernatant. So when we perform the centrifugation for the first time, then we will see that the plasma and the serum which will be obtained in the topmost layer, whereas the platelets, white blood cells and RBCs will be present in the pellet. So we will remove that supernatant containing the plasma and serum, and we will again centrifuge it. And with each round of centrifugation, more you can see that higher level of that sedimentation will be observed. So at last you will see that you will be able to separate the RBCs from the WBCs. So you will take the WBCs only, and then you will perform restriction digestion from the DNA that you have isolated from the WBCs. And then after restriction digestion, we will perform agarose gel electrophoresis, in which we will put the sample of the DNAs in different lanes, and we will allow it to move in the presence of electric charge, as we have previously discussed. Now the denatured DNA will move in those lanes, and we will observe different bands in that. So after the electrophoresis is completed, we will perform southern hybridization or southern transfer or southern blotting. These are three different names for the same technique. So hybridization is the technique in which we transfer the bands from one location to the another. So for example, in this we are transferring them from the agarose gel to the another gel, or we can say another sheet, that could be nitrocellulose membrane or nylon sheet. So in this way, by putting the weight over them, we will allow the bands to transfer from the gel to particular membrane, and then that membrane will be taken, and now you have got the single stranded DNA replica of a gel. So replica means exact similar patterns of the gel will be obtained on that. So you can obtain it either on the membrane or on the another gel. So now what you would do, you will add the probes, and then you will wash it, and you will observe it under the you will perform autoradiography to observe the bands, and at last you will obtain the bands, and you can see, and now you can compare. So on the basis of different DNAs you have used, you can compare the samples, and in one lane you can put the marker for the comparison. So marker is the band which has already the fragments that are present, and now with the another bands another sample that you have provided, you can compare between the fragments and fragment lengths. So this is very helpful. There are many applications of this analysis. So there are few which I have written in the right side, which means the disease diagnosis, genetic mapping, forensic science, genetically modified organisms, paternity testing, personal identification, plant breeding, characterization of genetic diversity, species identification, etc. So these all can be performed with the help of RFLP analysis. So this method is very promising and very effective to do all these diagnoses. And now we come to DNA fingerprinting and DNA profiling. So DNA fingerprinting, it's very common. We often heard this name again and again when it comes to the forensics or paternity testing. Then we hear it a lot, that DNA fingerprinting, and then we also know that DNA fingerprints of each human being are unique. So this is also helpful in distinguishing the individuals from one another. For example, if you obtain a DNA from the crime scene, then also you can by comparing its bands with the suspect's bands, you can identify that who is the criminal among the suspects. So in this way, there are two different terms which are often confused that what does it mean. One is DNA fingerprinting and one is DNA profiling. So they are not much different terms, they are only the subcategory of one another. So in DNA fingerprinting, the analysis of DNA is performed in order to identify the individuals, whereas in DNA profiling, the analysis of the individuals' DNA characteristics for the forensic studies. So when you are doing it for the forensics, then it is known as DNA profiling. You are creating a profile. And the next point is a molecular genetic method which allows the identification of individuals according to the unique patterns of DNA. So whenever you're going to perform the unique identification of an individual, then you perform DNA fingerprinting. But when you perform it for the forensic technique, and which is important for the criminal investigation as well as the paternal testing, or you can say parentage testing, when you want to identify the parents, then you perform DNA profiling in which you generate different profiles of the DNA and you compare them. So in DNA fingerprinting, we focus on the VNTRs, that is variable number of tandem repeats, including both mini satellites and microsatellites, whereas in DNA profiling we mainly focus on STRs, that are short tandem repeats, which are microsatellites. So these are nothing but these are the repeats. In micro and mini satellites, so they vary from one another in the size of the, or you can say in the number of nucleotides that are present in the repeats and number of times they repeat. So STRs and VNTRs, they are the repeats that could repeat in the repeats that could be present up to millions or trillion repeats that could be present. So these are multiple repeats, and micro means they can be dinucleotides that are repeating, and mini means if more than 2, means from 6 to 10, if they are the nucleotides that are repeating, that is a particular set of nucleotides that is repeating continuously, that is known as mini satellites. And micro means even lesser size of the repeat is present, then it is microsatellites. The techniques which we use for DNA fingerprinting are RFLP, AFLP and PCR, whereas for DNA profiling we use PCR. So as we have discussed about RFLP and PCR, so we know that PCR is a technique which is performed in vitro, whereas RFLP is a technique which is performed in vivo, and due to which the technique which is performed in vivo is very time consuming. Hence for the forensics we do not have that much time, so we use PCRs so that we could identify it quickly. And then and last point says that it is DNA fingerprinting is a cumbersome method with many steps, whereas DNA profiling is a simple process which can be automated as well. Okay, so I hope it is clear. So now let's move forward. Now let's see diagrammatically the DNA fingerprinting and DNA profiling. So in DNA fingerprinting, as I was talking earlier, that you use RFLP based techniques. So similarly in this, the restriction digestion will be performed, and similarly I've already discussed this method, that how southern blot is performed, in which after the digestion with the restriction enzymes you perform electrophoresis, and then you transfer the DNA onto the membrane, and then you hybridize it with the probe, and then the radioactive bands of different mobility you can observe. So this probing is generally done to identify the region of interest. If you want, then you can use this probe. Now let's look for the PCR based DNA profiling. So in PCR based profiling, you don't have to do restriction and digestion. You simply provides the DNA, you provides the forward reverse primers, the NTPs and the Taq polymerase, and then you repeat it for the multiple cycles so that you can amplify the DNA that is present with you. So now the difference that we will observe in the PCR based DNA profiling is that you look for the VNTRs. So not just VNTRs, but in them also you look for the microsatellites. So these are 3 different individuals, and this is the after electrophoresis, these are the bands you will obtain, and this specifies the number of repeats, that how many number of repeats are present, and on the basis of number of repeats you can identify that you can differentiate between all these 3 individuals. So it is based on the number of repeats, and it was based on the size of the fragments, the RFLP technique. So this is the difference, and why this PCR is faster, because it does not involve all these things such as southern hybridization, restriction digestion and others. So now let's solve this question, as we have discussed a lot about the fingerprinting, so this would be easier for you. So the question says, the DNA fingerprinting analysis shown below was performed for a family dispute in which 3 males, male 1 to 3, claim to be the biological father of the child offspring Examine the profile and find those males who cannot be the biological father of this child. So in this, by observing the DNA fingerprinting analysis, you have to tell which one among these males could never be the father of this offspring. So now these are the bands and these are the options. So you can observe this and you can answer this in the chat box. It is very easy. I believe everyone should answer for this question. Okay, so some are saying option B, male 1 and 3. Okay. Anyone has doubt in this? I think majority of students are answering option B, that is male 1 and 3. Okay, so now let's see. It's the correct option. So now why is it male 1 and 3? For the ones who don't know that why is it true? Because if you see the bands of mother and the offspring, then you see these bands are not present in the offspring, and the offspring will only show the bands which are inherited from his mother and his father. So that means any band that is present in offspring, it must be present either in mother or either in father. So here this band is unique for this offspring that is not present in mother, so it should be present in the father. So in male 1 this band is absent, in male 2 this band is present, in male 3 this band is absent. So from here only you can see that male 1 and 3 can never be father of the offspring because they lack that band, and this band is also missing in the mother, so it must be present in the father, and so male 2 is the biological father of this offspring. So hence which males are not his fathers? Male 1 and male 3. So option B is the correct option, and you were right. So now let's move further, and now this is another question, so which says, what are the differences in the specific regions of the DNA sequence called during the DNA fingerprinting? So this is saying when you perform DNA fingerprinting and you observe the differences in specific regions, so what are those regions called? As I have discussed with you this DNA fingerprinting and profiling, so I have told you that there are certain regions, because we know that among all the human beings, most of the genetic part is exactly same, but there are few regions that vary among all the individuals, and those regions are generally the repeats. Okay, so look at these options. One is non-repetitive DNA, another is repetitive DNA, third option is satellite DNA, and fourth option is histone DNA. So what do you think? What are the specific regions where differences are observed in the DNA sequence during fingerprinting? Okay, answer in your chat box. Okay, so many of the students have mentioned option B, and still there are certain students who says option B and C. They are confused between option B and C. Okay, so now let's see what will be the correct option. So the correct option is option B, that means repetitive DNA. So in DNA fingerprinting, the differences in the specific regions of DNA sequence are known as repetitive DNA. And in this part, a small structure of DNA is repeated many times, and these repetitive structures can be separated from the bulk genomic DNA, and this is done by observing the different peaks achieved by them during the density gradient centrifugation. So different peaks on the basis of number of repeats and on the size of the particular DNA sequence, the peaks are observed. So now the students who are confused between repetitive DNA and satellite DNA, so for their query I would answer that repetitive DNA is an umbrella term for different types of repeats, whereas satellite DNA is a subtype among the repetitive DNA, because repetitive DNA have multiple types. So for example, here I have shown you the repetitive DNA can be highly repetitive or middle repetitive. So in highly repetitive, it comes satellite DNA. Satellite DNA is very small, and why is it not the answer there? Because satellite DNA is predominantly present in the telomeres as well as centromere region. It is not distributed throughout the chromosome. But when we talk about the DNA fingerprinting, then we are not focusing on the terminal regions or the central part of the chromosome, but we are talking as a whole. So if to consider the entire chromosome or entire DNA that is present, so we cannot select satellite DNA here because the differences are also present at the other parts than the telomeres and centromeres. So in this way, repetitive DNA would be the correct option here, because we are talking in general. We are not focusing on a particular part of the chromosome. So hence, satellite DNA is not the correct option. Whereas if you look here, then another category is middle repetitive. That is, as compared to satellite DNA, the repeats will be lesser in number. So one is tandem repeats, another one is interspersed retrotransposons. So tandem means which are present with one another in the tandem manner. Okay, so whereas another one is interspersed, means that are distributed unevenly, and it's not necessarily to be present in the form of tandem. So you can say, means tandem can also be looked in the form as consecutive order, or the things that are arranged one after the another. So interspersed means it is spread out, it is not present one after the other. Whereas in tandem repeats, you can see multiple copy genes such as rRNA genes, mini satellites you have heard earlier also, which are the VNTRs, and microsatellites, that are the dinucleotides. And in sign and line, that are the small interspersed nuclear elements and long interspersed nuclear elements. These are their examples, ALU and L1. So these are the retrotransposons that are interspersed. So these are the different things, different repetitive DNAs, which uniquely signifies an individual and which is helpful in distinguishing one individual from the another. And these are the reasons behind the unique DNA fingerprint of an individual. I hope it is clear. So if there is no further doubt, we'll move forward to another question which is very similar to the previous question. Now this question says that what are the small peaks achieved by the repetitive DNA during the density gradient centrifugation process of DNA fingerprinting known as. So now you have to tell that what are the small peaks that are achieved after the repetitive DNA during the density gradient centrifugation is known. So whenever you perform DNA fingerprinting, in the previous also I have discussed with you that whenever you take the DNA and you're observing the fingerprints, so you whenever you perform density gradient centrifugation, then you observe different peaks based on the size. So here they are saying small peaks. So we have already discussed about the repetitive DNA. So first option is non-repetitive DNA, option B is TRUF, option C is satellite DNA, and option D is histone DNA. So can someone answer? Okay. So many students are confused. Okay, no problem. We'll see. So first of all, you can reject few options which you know that they would never be true. For example, this non-repetitive DNA. So this can never be true, because in DNA fingerprinting we are focusing on the repetitive DNA, because those are the only sites which helps in distinguishing the individuals. So TRUF can also not be the true option, because TRUFs are not the smallest peaks that you see. They are not the peaks. Then comes the option D, that is histone DNA. So histone DNA, why would you look for the histone? Because histone is a protein that helps in DNA to wrap around each other. So they do not form the small peaks, because in this you are entirely focusing on the DNA. You have obtained the DNA, not the histone with it. So now we are left with only option C, that is satellite DNA. And I have told you that when we talk about the repetitive DNA, then we see that these are highly repetitive and these are satellite DNAs. And for your information, I would tell you that satellite DNAs are very smaller in the sequence size. So hence you will look for the small peaks of these satellite DNAs. So genomic DNA forms a high peak in the density gradient centrifugation. So the genomic DNA will form high peaks, whereas the various small peaks which are formed are collectively called as satellite DNA, because they are obtained from the centromeres as well as telomere regions. So they will show the smaller peaks. So hence they are regarded as satellite DNA. Now let's move to another question. It says specific biomolecules which show easily detectable differences among different strains of a species or among different species is termed as. So whenever you compare different species, so what do you think, what are the specific biomolecules which are helpful for detecting the differences among different strains? For example, if you are comparing two different strains of bacteria, or if you are comparing two different species of another organism, there are two different species, genus is same but the species are different. So what do you think, what is that particular biomolecule called which is helping you out in detecting this difference? Option A says DNA fingerprinting, option B says molecular markers, option C says molecular scissors, and option D says RFLP. So now answer this. What do you think? So option 1 says DNA fingerprinting, and you know that DNA fingerprinting is the method that you use, it is not a biomolecule. Now option D is RFLP. So RFLP is also a method that is based on the restriction fragment length polymorphism. Then option C is molecular scissors. So restriction enzymes are also regarded as molecular scissors because they are producing the cut, and restriction enzymes are the external agents. So they are not helping you out in identifying the species. So we are only left with a single option, that is molecular markers. So yeah, right. So with this you must know that what are molecular markers. So these are defined as any DNA sequence which shows polymorphism and can be detected using a molecular technique. So in this way, these molecular markers are present within the DNA sequence. And what are the different types of molecular markers that are present? So for your information, I have attached these also. So there are different techniques, not just RFLP, but other techniques are also present. So in this way, you can see one is RFLP markers. So RFLP markers means, for RFLP we require the variation in the restriction enzyme cleavage sites. So whenever in a sequence, if the site of cleavage is present at some other positions, or if it is for example, if an individual has undergone some mutation in some recognition sequence, so in that case the number of cleavage sites will vary. For example, in the normal situation, if there are 3 restriction digestion sites present for EcoR1 enzyme, and now an individual is suffering from a particular disease in which his sequence has undergone some change, due to which one of the cleavage site got modified. So now it's no longer coding for the cleavage site. So now there is only 2 cleavage sites present. So now you can see that there will be difference in the RFLP pattern, due to which you will see there will be different bands observed. So in this way you can distinguish between the two species. And now there are another techniques such as RAPD and AFLP, as well as mini satellite markers, micro satellite markers, and SNPs, that are single nucleotide polymorphisms. So these all molecular markers helps in distinguishing among the species, and these are different techniques that you can use: RFLP, RAPD, and AFLP. So now let's move to the another question. So now the question says, the sequences of satellite DNA do not code for proteins. So now you have to tell that either the sequence of satellite DNA code for protein or not. Is it true or is it false? So here they are saying that they do not code for proteins. What do you think? We already discussed about the satellite DNAs. What are these? These are some kind of repeat sequences of nucleotides that could be present in million number of times in the DNA. So what do you think? Many people are quite confused. Okay. Some people are saying option A is right. Okay, let's see. So now the correct option is option A, which says it's true. Yeah, so the sequence of the satellite DNA do not code for proteins. As I've already told you that these are the DNA sequences that are specifically present in the telomere regions as well as in the centromere regions. And I've also told you that in the telomere regions, these are generally contain the part of the DNA which generally do not code for any of the proteins. And you must know that with each round of replication there occurs telomere shortening, and to avoid this telomere shortening in eukaryotic organisms, there is an enzyme that is telomerase, which helps the eukaryotic chromosome from the telomere shortening. But with aging, this enzyme reduces, and hence telomere shortening happens. So somebody in the chat has asked me that in the Y chromosome they have heard that there is the cases of telomere shortening and loss of gene. So as I am saying, the telomere part of the chromosome is not associated with the coding part. But what happens is that with each round of replication, as telomere shortening happens, so there is a term which is known as Hayflick's limit, which says that it is a limit till which even if telomere shortening occurs, but it does not involve the loss of genes. And after Hayflick's limit, if further telomere shortening happens, then it can involve the loss of genes. So if someone has asked about the Y chromosome, that it is undergoing loss of gene from the telomere ends, so the possible reason behind this could be that maybe the telomere has shortened enough, it has crossed the Hayflick's limit, so now with each round of further replication it is leading to the loss of coding part, or the part that is coding for the gene. So that could be the possible reason behind this. So now let's look into its explanation. So it says that the small peaks formed by the DNA when kept in the density gradient centrifugation are called as satellite DNA. And these DNA sequences do not code for proteins. On the contrary, they form a large portion in the genome of humans. So it is saying although the satellite DNA constitute the larger part, but it does not code for the protein. And these are the repeat regions that are present, and their size or the sequence is very small, that is continuously repeating. So still it is constituting the larger part, but it does not code for any protein. So that's true. Now let's move to the next question. So the question says, how does polymorphism arise? So I hope you are aware about this term polymorphism, and the options says option A is mutations, option B says recombination, option C says diploidy, and option D says haploidy. So what do you think? So when we talk about the polymorphism, then polymorphism means if a particular thing can exist in multiple forms. For example, in human being, different individuals are almost similar, but their genomic content can vary in certain amount. It can either vary in a single nucleotide, or it can vary in its sequence at a particular site. Due to mutations it can vary, or there are different reasons behind this polymorphism. So what do you think? We can also view polymorphism as occurrence or two or more clearly different forms or different morphs. So it is also referred to as alternative phenotypes in the population of species. So that is all about the polymorphism. So what do you think? Okay, many people are saying mutations. They might have heard as I was explaining it. Okay. So let's see without wasting further time. Yeah, it's correct. Answer is option A, that is mutations. So yeah, mutations is a possible reason behind the polymorphism. So what it says that polymorphism arises due to the mutations. It can also be known as the discontinuous variation that is seen among the individuals of species. So as I have told you that among the individual of species it is observed, and mutations are discontinuous. It means it is not continuous. For example, you don't know how much variation you will see, and it is also not fixed, that this number is not fixed, it can vary, so it can be discontinuous, right? So now somebody was asking me the question when I was explaining the pedigree. So now I have taken this from the your assignment 2. So the question which I have solved, it was to calculate the penetrance. But many people were confused, and their confusion was that in this question they have to calculate the minimum number of you can say the organisms that are showing the genotype but not the phenotype. Okay, so see the pedigree, its correct answer was option 3, that is 2. And it is here they are saying that the minimum number of people who have the disease linked genotype but not the phenotype are 2. So now you're confused that why its answer is 2. So the question which I solved, so in that we had only 3 generations. So here they have added one more generation, that is generation 4. So now let's see. These black colored are affected individuals, and that means these are all the penetrants. So now how to identify the known penetrants? So first of all, as I've earlier discussed also, that whenever we heard about the penetrance or incomplete penetrance, so incomplete penetrance is a feature of autosomal dominant inheritance pattern. So what is autosomal dominant? Autosomal dominant inheritance means from either of the parent, even if a single dominant allele is present, then it will cause an effect. And when the recessive alleles will be present, then the individual will be unaffected. So let's see from the generation 1. So the father was affected and mother was unaffected. So if we have to write its genotype, then it might have capital A, small a, and the unaffected individuals will definitely have the recessive alleles. So it's small a, and it could also be double capital A. But now let's look at its offsprings. So it says that 2 of its daughters were normal and 1 of the son was affected, which means that if it was having both the dominant alleles, then it's all the progeny should be affected. But since we see that 2 unaffected daughters are present, which means its genotype should be capital A, small a, 1 dominant allele and 1 recessive allele must be present in this. So now let's see the generation 2. So in generation 2, if we see from here 1 and 2 of generation 2, then they are unaffected, which means for any individual to be unaffected, both the recessive alleles must be present. So small a, small a, small a, small a. So now we see that their offspring is affected, which is not possible. So that means in this case, the 1, it might have the genotype of the disease allele, but it is not showing the phenotype. So it must be capital A, small a. So due to which it is showing incomplete penetrance for the disease trait. Okay, so in this case 1 is this incomplete penetrant is present, which has the genotype but not the phenotype. Now let's move here. So here it is showing that father was affected, mother was unaffected, and their children, both their children are unaffected. So if this has a capital A, small a, and this has small a, then there are chances that their offsprings could be unaffected, because this parent is only transferring his a recessive allele, due to which there is no disease observed. So which means there is nothing like incomplete penetrance here. Now let's move here. So here also the case is same, that both the parents are unaffected but one of their offspring is affected, and which is not possible generally. It only happens if their parent is hiding the phenotype, and most probably this mother should be the showing the incomplete penetrance, because its parent was having the disease trait. So which means it is having the genotype for the disease, but it is not showing the phenotype. That means it has undergone the incomplete penetrance for that trait. So in this way, here we see that there are 2 individuals which has the genotype but not the phenotype. Now let's observe generation 3 to check that are there further cases which could have the incomplete penetrance. So here also, as we see that one of the parent was affected and one of their children is affected, so it's quite clear. So this individual has passed the disease trait to here, and similarly these 2 haven't married, there is no cross between them. So okay, so now how many individuals have the genotype but not the phenotype? There are 2. One is this second generation individual 1 and second generation individual 5. These are the 2 daughters. So they are showing the incomplete penetrance. So I hope now this concept is clear. So now let's move to the another question. So many of the people were having the doubt regarding what of the techniques which we can use for identifying the disorders. So I have collected few information from the online resources which says that these are the diagnostic tests which you can perform, and these are the type of disorders which are generally associated. So in this type 1, here is the chromosomal disorders. So whenever you have to identify the chromosomal disorders, then the diagnostic tests which are generally performed involves the chromosomal analysis. So in this you perform the analysis of the chromosomes, you check for the karyotypes, And when it comes to single gene disorders, whenever a single gene is associated with certain disease, then how you can track those diseases? So you can perform pedigree analysis, you can perform clinical examinations, you can perform biochemical analysis, and you can also perform the deoxyribonucleic acid analysis. Then comes multifactorial disorders. So the disorders which involve multiple factors. So for that, the diagnostic tests which could be performed includes clinical examination, biochemical analysis, deoxyribonucleic acid analysis, and other investigations such as imaging, functional studies, for example, you can perform for these. Then comes mitochondrial disorders. So there are certain disorders that are generally occur due to the disorders in the mitochondria. So for that, you can perform pedigree analysis. You can also perform the clinical examinations and deoxyribonucleic acid analysis testing. So these are the few diagnostic tests which you perform when you have the mitochondrial disorders. Then comes somatic cell genetic disorders. So when the disorders are occurring in the somatic cells, so then you can perform histopathology, deoxyribonucleic acid analysis, and chromosomal analysis. So these are few of the diagnostic tests which you can perform, because till now we have studied about the pedigree analysis, and we have also looked into the DNA fingerprinting, the RNA cloning, DNA cloning, and hybridisation techniques. So other than that, these are few of the diagnostic tests which are generally performed depending on the what type of disorder is present. So on the type of disorder, we would select among these that which diagnostic test we want to perform. These are few of the other investigations in which it says that in routine, generally the tests or investigations which are performed are chromosomes, FISH for microdeletions, and we perform DNA tests for single gene disorders. Then there are newer diagnostic aids also available, which involves telomere studies and 1 to 3 megabase interstitial deletion screenings and microarray studies, which involves the silicon chip based. So these are few of the more diagnostic methods or techniques which can be used. Then comes prenatal diagnosis. So for the prenatal diagnosis, for the child which is not born yet, so for that, ultrasound examination which involves major structural abnormalities of nervous system, which is anencephaly, spina bifida, so hydrocephalus, obstruction of urinary tract, failure of kidneys to develop, failure of limbs to form normally. So whenever an individual is not born yet, still we can go for the ultrasound examination to check that whether if that individual born, so that will be fine or not. So before that, we can perform this diagnosis. And then comes fetal DNA analysis. So we can determine the biochemical abnormalities by analysis of the DNA of the fetal cell, for which we can perform amniocentesis and chorionic villus sampling. So these are generally performed to analyze the DNA of the fetus. So these are different methods or different techniques which, if you want to have idea, so you should have that these are the tests which are generally performed for the diagnostics related to disease. So now in the next session we will talk about week 3 topics, and we will perform few MCQs which are related to week 3, and by that time I guess your week 3 assignment results will also be out, so we will be discussing much about them. And along with that, if you have any problems related to session 1 or session 2 or week 1 or week 2 content, then also you can ask anytime. And for the next session, you can come up with the problems related to week 3 content, which generally involves the mutation and instability of human DNA. In that, we will be looking into the mutations. We will also again go through the polymorphism in depth. We will look for the pathogenic mutations. And you must know that not all the mutations are pathogenic. Certain mutations are also good mutations. They help in gain of function, or they can help in the individual for his survival. So not all the mutations are harmful, but we will focus on the pathogenic mutations. We will look for the repeat expansions. There are certain certain disorders that are associated with the DNA repeats. So whenever this repeats expands, then it causes abnormality or cause some disorder, some disease. So we will also check for those repeat expansions, that which are the disease which are related to these. We will also look for the molecular pathology, which will be containing the types of mutations that are responsible, and we will also look for the animal models for the human disease. So these will be the topics for the next session. So I'll just take few doubts. If you have, you can write in the chat box, and then just we'll end the session. Okay, so one of the doubt which I have received so far was about the question which I discussed in the beginning about the genomic DNA when it converts to the RNA. Then many people have the confusion. Somebody was saying that the parts, the exons and the introns, so they were confused that when alternative splicing happens, then different choice of exons are being selected and certain exons they are skipped. So people are saying that those exons are regarded as introns as they are not coding for the particular transcript. So for your information, I would tell you that exons, as we say, exons are the coding part and introns are the non-coding parts. And if we talk about their functional relevance, then they both are equally relevant because the non-coding part has some regulatory role, whereas the exons, they constitute the protein. And there was also a doubt that not all the part of exon is contributing to the protein. So yeah, it was right, because when we discussed our first MCQ, then we saw that at the 5' end there is a cap, and after the cap there is a region till AUG, that is the start codon from where the protein translation begins. So before that, the part is known as 5' UTR, and that part is untranslated region and it comes from the exon. So yeah, the question means the thing that not every part of exon is coding for the protein. There are certain part of the exon which can present as a UTR, that is untranslated region, but that part will never be called as an intron. So whenever we say the exons are the coding part, so we means that they are the coding in terms of transcription, they transcribes, and further they means the exons that are present at the 5 terminal and 3 terminal, means that are present before AUG and after stop codon, they are referred to as UTRs. So in this way, means if we talk about the middle exons, then they are always coding for the protein, and introns will never code for the protein. And when we will talk about different cases of splicing, so we know that there are different types of splicing. One of them is alternative splicing, in which the different combination of exons has been selected. So there could be different type of splicing, such as in one splicing event exon is skipped, whereas in another splicing event exon is mutually exclusive exons might be present, in which means mutually exclusive exons are the exons which can never occur together, but they occur individually in two different transcripts. And then there are certain cases where there is intron retention. So intron retention events are also observed, in which due to the missing of the splice site, the splicing does not occur and intron get retained within the two exons, and in that way it enters the transcript. So those are few exceptions where we see that introns enters the RNA and it does not get spliced out, because during the splicing every intron has to be removed, but in certain situations intron get retained because its splice site remain hidden and spliceosome could not splice it out. So there are certain cases where we see that intron get retained and then further it undergoes translation. So in those cases you can say that intron is getting translated, but not in all cases. In certain situations, after the retainment of the intron within two exons, it could also lead to the premature termination of a protein, and not always. It can certainly sometimes code for the protein also. But if we talk in the general sense, then we will say that introns will never they are not self-sufficient to code for any protein, so they will never code for the protein. But yeah, if we want to talk about exceptions, then we can say during the splicing, even if intron retention happens, then in certain cases, as exons are present in both the terminal of that intron, so due to that exons, that particular intron is able to code for the protein. So those are the exceptions. So I hope now this doubt is clear, and as I have no diagram to show here, so I hope means you would be able to see. And if you have any confusion, then during listening to this lecture you can open up the Google, or you can see somewhere where it's mentioned where the DNA is getting converted to RNA and how introns and exons are aligned, so you'll be better able to understand that what I am saying. So I hope I have solved your doubt, and even if you still have the doubt, then in next session you can ask me, and means I'll try to bring up the figure so that I can explain you it in a better way. So till then, see you in the next session. Then I'm just ending this session. Okay, then see you. Thank you so much for listening to me. Goodbye, and see you on next Tuesday.